Okay, well, thank you very much for coming tonight. It's great to see such a good turnout. It's actually a real pleasure for me to be invited to give one of these talks. I give a lot of presentations to a hardcore scientific audience that focuses very much on the nitty-gritty detail of our research. So it's actually really nice to have the opportunity to step back a bit and look at the broader picture of what we're actually trying to achieve and why. So as you just heard, I'm a laboratory scientist, I'm not a medical doctor. So we're coming at this very much from the point of view of trying to understand why type 1 diabetes develops. The idea being that if we can understand the pathways that set diabetes in motion, then we know what it is we have to try and interrupt to stop it. So what I'd like to do over the next 45 minutes or so is talk to you a little bit about some general background around type 1 diabetes, talk about what we know already about the disease process, and then tell you a little bit about what research my team is doing to try and push the field forward. So type 1 diabetes is the form of diabetes that typically arises in young people. It used to be called juvenile diabetes for that reason. The average age of onset is around 12, but it can develop at any age. There are around 400,000 people in the UK living with type 1 diabetes, and over 29,000 of these are children. We hear a lot in the news about the increase in obesity and type 2 diabetes, but what many people don't really appreciate is that type 1 diabetes is also rising very rapidly. So we're looking at the incidence growing by around 3% per year. And if we focus on preschool children, so children that are under the age of 5, the incidence seems to be increasing at a rate of 5% per year. Just last year, it was revealed that the UK now has the 5th highest rate of type 1 diabetes diagnosis. So this is clearly a healthcare problem that's escalating very rapidly. And it's not just in the UK. This world map shows the number of new cases of type 1 diabetes per year, and the darker the green, the higher the number of cases. And you can see that it's not just the UK that's dark green, but also Scandinavia, Saudi Arabia, North America, and Australia are all seeing high numbers of new cases. This is the listing down here. It's not really clear why the incidence is increasing so rapidly and why we're seeing such variation between different countries. There are a large number of theories that have been put forward. It's been suggested, for example, that higher latitudes confer a greater risk, possibly because of lower vitamin D levels being obtained from sunlight. It's also been suggested that the affluence of the country plays a role. So the more well-off countries generally have a higher incidence of type 1 diabetes. Now, whether this is because of something that these countries have or because of something that the countries don't have, the absence of a particular infection or parasite, for example, isn't really clear. Obviously, the genetics of the people that live in these different countries is also different. So it's actually hard to know whether the incidence in a particular place reflects the genetics of the people that live there or the environment of that place itself. And it's actually really tricky to unpick whether a particular trend is due to genes or to environment. So to illustrate this, if we look at men who play rugby, as I like to do, we can see clear evidence that rugby success can run in families. There are many, many examples of brothers, both of whom are successful rugby players. So we've got Tom and Ben Youngs, Dylan and Stefan Armitage, Mako and Billy Vunipola, And there are many other examples, the Hastings brothers, the Lamonts, the Underwoods. Is this because the brothers share genes that make them good at playing rugby? Or is it because of the environment these guys were brought up in? The amount of parental support, their diet, where they lived, and so on? Or is it actually a bit of both? And it's actually very hard to unpick these two effects of nature versus nurture. With type 1 diabetes, we know that genetics clearly plays a role, and we know that there's also an environmental contribution. But exactly what these environmental factors are and how they interact with the genes that we believe are involved in diabetes isn't really well understood. So what do we know? Well, we know that type 1 diabetes is caused by an immune-mediated attack on the cells that produce insulin in the pancreas. So here's your pancreas. It lies here within the abdomen. You can probably see the pancreatic duct running down the middle of it, and this carries the enzymes to the gut to help with digestion. And within this pancreas tissue, there are small clusters of cells called islets of Langerhans, after the person that found them. And each of these islets is about the size of a grain of salt. And within these islets are the beta cells that are shown here in orange, and it's these beta cells that are making insulin. And it's these beta cells that are targeted and destroyed by the immune system in type 1 diabetes. When we can't make enough insulin, this is when the symptoms of diabetes occur. So we need insulin in order to allow cells to take up sugar and allow them to make energy. So if there's not enough insulin around, then cellular metabolism fails because the cells aren't able to make energy. And then because the cells aren't taking the sugar up, we end up with very high levels of sugar left in the blood. And this causes a number of problems. It causes damage to nerves, eyes, kidneys, and blood vessels, leading to the complications that are associated with type 1 diabetes. So lack of insulin is the fundamental problem in type 1 diabetes. And the discovery of insulin in the 1920s, as we heard about, really revolutionized our ability to treat type 1 diabetes. And just to illustrate this, I want to tell you a true story about a man called Mr. Brostoff. Now, Mr. Brostoff was diagnosed with type 1 diabetes in 1909, and he was 29 years old at the time. And this was well before insulin had been discovered. At that time, diagnosis with type 1 was really a death sentence, and he was told that he had limited life expectancy and that he needed to make arrangements. So at this time, life expectancy after a diagnosis of type 1 was less than 4 years, and in fact it was quite common to die within a few months of diagnosis. Mr. Brostoff was put on an incredibly strict diet and was only taking in 1,000 calories per day, and this was regularly bringing him to the brink of collapse. Incredibly, he managed to survive 14 years like this, although he was reduced to skeletal form. Then, in 1921, the Canadian group of scientists led by Frederick Banting discovered insulin, and Mr. Brostoff was one of the first people in the UK to receive it. As soon as he started taking insulin, he was able to increase his calorific intake, his weight improved, and he lived to a ripe old age of 88. So this is an amazing illustration of how life-changing this discovery of insulin actually was. So this is Frederick Banting here on the right with his medical student Charles Best. This was the year that they discovered insulin at the University of Toronto. They're standing on the roof of the medical school. It's not really clear why. And the way they made their discovery is that they actually took the pancreas out of a dog and prepared a material from it that they called iletin, after the pancreatic islets. And they found that if they injected this substance into a dog that had diabetes, the symptoms disappeared. Health and safety was far less strict in those days, so excited by their discovery, they promptly injected it into themselves. They felt a bit faint, but were otherwise okay. So with the safety testing out of the way, they then injected it into the 14-year-old boy that we heard about earlier, Leonard Thompson, who was very ill at the time. He'd actually been diagnosed 3 years previously, so he was near death. But after injection with this iletin, he rapidly recovered. So again, this is another story emphasizing how life-changing the insulin discovery was. So we now have much better forms of insulin and far better ways of delivering it. But although this is a way of managing diabetes, it's not actually a cure. We're not there yet. Even though we have insulin, life with type 1 diabetes is still challenging. And I think this is very nicely summed up by some statements on the JDRF website. I've taken the liberty of selecting a few to show today. So these are written by people with type 1 diabetes. So this one here talks about the need to be a mathematician, physician, personal trainer, and dietitian all rolled into one in order to factor constantly how much insulin and food to take. This one talks about never getting to relax and forget about food, or worrying about the impact of exercise, stress, a cold, sunburn, etc. And this one is written by an 8-year-old who talks about being different from the other kids because of having to check blood sugar levels so many times a day and measure everything that they eat. So having insulin is a big step forward, but we've not solved the problem of type 1 diabetes yet, and we still need to do more. So to tackle the root cause of type 1 diabetes, rather than just manage the condition, we need to understand it. And this requires a long-term investment in high-quality research. So I think there are no quick answers. We need careful, high-quality science that over a time scale of years will uncover the mechanisms that actually drive diabetes and provide us with the clues that we need in order to stop it. So how do we go about doing this from a research perspective? Well, let's start with what we know. We know that diabetes is caused by the immune system, and we know that the cells of the immune system travel around the body in the blood. So let's start by looking at some blood cells. So this is a photograph of some of the cells from the blood. On the left, we've got a red blood cell. These are the cells that carry oxygen around the body. In the middle here, this little cell is a platelet, and the platelets are the ones that are involved in blood clotting, so prevent us from bleeding to death every time we cut ourselves. And on the right, this fluffy cell here, this is a white blood cell. And it's the white blood cells that are responsible for our immune-mediated defense and are also responsible for causing type 1 diabetes. So this immune defense function is absolutely critical to our survival. Our environment is full of microbes, different types of viruses, bacteria, fungi, parasites, etc., all of which are continually trying to invade us and could give us diseases if we didn't have a strong, healthy immune system to fight them off. some of you may have heard of the boy in the bubble, who was a famous case of a child who was born without a functioning immune system. And this boy had to live out his short life inside a sterile bubble, because any exposure to the outside environment would have meant death from microbial attack that he had no way of defending himself against. So the immune system is absolutely critical for us to survive. However, there's another side to the immune system, and in fact we often talk about it as a double-edged sword, because even though it's absolutely essential to defend us from infection, it can also have side effects that can cause us harm. So if immune responses, rather than being targeted against invading microbes, instead are targeted against our own tissues, we can develop autoimmune diseases. And the precise symptoms depend on which tissue is being targeted. So the joints in rheumatoid arthritis, the pancreas in diabetes, the nervous system in multiple sclerosis, and so on. Another issue is if the immune system decides to launch an attack against something that isn't dangerous, pollen or grass or cat fur. In these kinds of situations, we get the symptoms of allergy. So the immune system is essentially a powerful weapons system to target invading microbes, but if this targeting isn't accurate, it can cause collateral damage and it can actually do us harm. So back to the white blood cells that I told you are the ones that are responsible for immune defense, but also responsible for type 1 diabetes. There are actually a number of different types of white blood cell, some of which are shown here. And all of these different cells play a different role in the immune system. If we get attacked by bacteria, neutrophils are very good at ambushing them, getting them from either side and actually wrapping arms around them and physically engulfing the bacteria and killing them. If we get attacked by parasitic worms, these are typically too big to engulf, so instead we've got eosinophils that sit on them and cluster bomb them. The cell that's actually responsible for causing type 1 diabetes is the T cell, this cell down here. And T cells are essentially the snipers of the immune system. So they're very specific. They choose their target carefully, lock onto it, and home in on that particular target to attack. So what happens in type 1 diabetes is that these T cells go to the pancreas and they infiltrate these pancreatic islets, and they lock onto and destroy these orange beta cells that are making the insulin. So this is how it looks in real life. This is a section of pancreas tissue stained brown for insulin and blue for T cells. So this is a pancreatic islet here, and the brown cells are the beta cells making the insulin. And you can see here some T cells in blue starting to infiltrate that islet and nibble away at the edges of the beta cells. And then on the far right, you can see an islet that's now been virtually destroyed by the immune response, and there are only a few insulin-positive cells remaining. So why do we have these T cells that attack pancreatic islets? To answer this question, we need to talk a little bit about how we generate T cells. So the way it works is that each of our T cells recognizes one specific shape. So it's very much like the lock and key idea. So this T cell recognizes a shape on this particular microbe. This T cell is different. It recognizes something on this microbe, and so on and so on. And the problem is there are a huge number of different microbes, and they're changing all the time. So the T cells basically need to be able to recognize absolutely anything in order to defend us against what we don't know we're going to be attacked by. So essentially, T cells have to expect the unexpected. And of course, the problem with having T cells that are such a broad repertoire, that have the capacity to recognize everything, is the problem of having some T cells within that repertoire that recognize parts of ourselves and could give rise to autoimmunity. So the way that we try to get around this problem is, as our T cells develop, we make them go to school, we make them have an education process to try and tell them how to behave in such a way that won't give us disease. And this education process happens in the thymus gland. This is the thymus gland here, it overlays the heart. And our T cell precursors migrate from the bone marrow to the thymus, where they undergo this education process. And two important things happen during this thymic education process for our T cells. The first is that some of the T cells that recognize parts of our own body are weeded out and killed. So here we've got a nice broad repertoire of T cells, some of which recognize foreign microbes, but some of which recognize self. And what happens in this first step is that some of the T cells that recognize self are destroyed. So we've essentially skewed the repertoire of T cells away from recognizing self and towards recognizing foreign microbes. But you'll note that this process isn't complete. We still have some T cells left. This one here, for example, recognizes a pancreatic islet. So even though this one that recognizes a joint has been destroyed, we still have this potentially dangerous T cell left. So this is a skewing of the repertoire rather than a black and white decision. The second important thing that happens during this thymic education process is that some of the T cells that recognize self are turned into regulatory T cells, or Tregs. Now, these regulatory T cells have immune inhibitory function, so these cells actively inhibit autoimmunity. So during this thymic education process, we're getting rid of some of the T cells that could be dangerous, some of the cells that recognize self, and we're also generating a whole group of regulatory T cells that have inhibitory function and can downregulate immune responses. So essentially what we're left with in our mature T cell repertoire is a very fine balance. We have a small number of T cells that could potentially be dangerous and could cause autoimmune diseases by attacking parts of our own body. But we also have these regulatory T cells that act like the police force of the immune system, and they're actively keeping these dangerous guys in check. And virtually everything that we do in my lab revolves around trying to understand and manipulate this balance. So can we conceive of interventions that would inhibit these dangerous T cells but not impact these regulatory cells? Can we do things that might boost the Treg population and skew the balance towards regulation and away from disease? So within this conceptual framework, there are a large number of different research projects with different complex experimental approaches, but this is the fundamental basis of what we're trying to understand and what we're trying to manipulate. So let's just look at what I've told you so far. I've told you that type 1 diabetes develops because of an immune-mediated attack on the insulin-producing cells in the islets, these beta cells within the islet of Langerhans, and that white blood cells called T-cells are responsible for initiating this attack. There are a number of other types of immune cell that get involved and join in later, but we think it's the T-cells that are the driving force to really initiate this response. I've told you that we all have T-cells that have the capability of causing autoimmunity. We all have some self-reactive T-cells in our bloodstream. But whether we actually develop autoimmune diseases or not depends on genetic influences and environmental influences and multiple different inputs. And at the cellular level, what we think this comes down to is a balance between the T cells that have the potential to cause autoimmunity and the Tregs that actively hold these guys in check and inhibit autoimmunity. So how do we actually go about exploring this balance in the lab? Well, we use a very wide range of research techniques and research methods, from animal models to cell culture assays, gene expression analysis, cell biological approaches, analysis of blood and tissue samples from patients. And what I wanted to just tell you about this evening is just one research project that we've been doing as an illustration of the kind of things that we can do. And this research project revolves around asking the question of what are these T cells that are attacking the pancreatic islets actually like? What genes are they expressing? What factors are they producing? What is it that they're actually doing to cause damage? How can we find out more about these T cells? And the first principle to understand in this research project is that immune responses start in local lymph nodes. Lymph nodes are these little blue blobs here on the diagram, and these are little bean-like structures where the cells of the immune system come together to interact and share information. And you've probably felt your lymph nodes before. If you've had a sore throat and you said, oh, my glands are up, you can feel swelling. That's actually because your lymph nodes are swelling up because an immune response is being initiated in them. And the way it works is that every organ in the body has lymph nodes attached to it. And the decision of whether to mount an immune response in that particular organ is made in those local lymph nodes. So the pancreas has its own lymph nodes, these blue blobs down here. And the decision of whether to mount an immune response in the pancreas is taken in the pancreatic lymph nodes. So if we just home in on one of these lymph nodes and look at it a little bit more closely, this is a lymph node here down on the left, and we've cut it in two down the middle so we can see the cells in there. And then this is pancreas up here on the right. And what actually happens is that cells, material, fragments, bits from the pancreas all the time are being trafficked, being brought down to the pancreatic lymph node. And then the T cells in the pancreatic lymph node are taking the decision whether to mount an immune response or not. And ideally, if what is draining down from the pancreas is part of a microbe, part of an infectious agent, then we want the immune response to be initiated. But if it's just a normal pancreas cell, just a bit of islet, we don't. But this decision-making process isn't 100% accurate. So if the T cells do decide to mount an immune response to pancreatic tissue, what happens is they go from being resting T cells, naive T cells that are these small, round purple cells that aren't really doing much, to being activated angry T cells, so these red cells here. And then what happens when those T cells are activated is that they come out of the pancreatic lymph node and they travel to the pancreas and they start to destroy the islets. So these T cells that we were seeing nibbling away on the pancreatic islets are there because of a decision that was made back in the pancreatic lymph node here. So what we've been able to do as part of this project is we've been able to get at pancreatic lymph nodes using a mouse model of diabetes and purify these islet reactive T cells from the rest of the T cells within the pancreatic lymph node. And we've been able to do this using a high-speed cell sorting machine. And what this allows us to do is to isolate individual cells based on their characteristics so that we can then study that population of cells. And the way the machine works is that all of the pancreatic lymph node cells are passed through a laser beam, and then they're characterized, and the machine decides very quickly whether this is one of the cells that we want to study or whether it's not. So if it's not one of the cells that we're interested in, it gets passed into a waste tube. But if it's one of these rare cells that we want to study, what the machine does is it applies a charge to the droplet that contains that single cell, and this allows that droplet to be deflected as it passes these voltage plates into a different collection tube. So what we're able to end up with is a tube that's full of these very rare cells that we want to characterize and study. And what we've been able to do is then look in depth at these particular T cells to analyze what genes that they're expressing. So we now have a list of genetic characteristics for these T cells that initiate the diabetes process. And this has given us some important clues about really what kind of T cells these are and what they might be doing. And what we're doing at the moment, actually, in collaboration with my clinical colleague, Miranda Rosenthal, who's one of the diabetes consultants here, is we're now looking at T cells in the blood of people with diabetes to see if we can detect the gene signature of these cells that are responsible for initiating disease. So now we have some more information about these disease-inducing T cells. Can this help us to understand a bit more the balance between the bad T cells and the good T cells, if you like? One of the gene products that we actually found coming up in these T cells encoded a cytokine or a soluble factor called interleukin 21 or IL-21. So we've been interested to study this a little bit more in culture assays to try and find out what this does to the immune response. And we've had some very interesting findings that suggest that IL-21 interferes with immune regulation. So we've talked about how these regulatory T cells are able to suppress the disease-inducing T cells. So these are the police force that are inhibitory. And you can see this on the assay over here on the right. If you look at the black bars, what we're doing here is, as you move to the right of the assay, we're adding more of these regulatory T cells, and you can see that they're suppressing the response of these conventional T cells. So it's an in vitro suppression assay to measure the function of these regulators. But what we've found is if we put this soluble factor, IL-21, into these assays, then the Tregs are no longer able to suppress the responses of these conventional T cells, suggesting that soluble factors that are being made by the T cells that respond to pancreatic islets could be interfering with the natural suppression mechanisms that normally hold this response in check. So I've told you that everybody has T cells that can recognize self, so we all have the potential to develop autoimmune diseases. but that these T cells are normally tightly regulated and are really held in check by the regulatory T cells. And what this research project that I've told you about is really suggesting is that some of the factors produced by T cells as they respond to the pancreatic islets could be disturbing this balance between the regulators and the disease inducers. So this is something that we're keen to study further. So the main message of the talk is that the key to life is immune balance. And as I say, we're very keen now to build on the progress that we've made and probe further what's happening with this immune balance in people with type 1 diabetes. So as I mentioned, this research that we're doing is taking place in the new UCL Institute for Immunity and Transplantation. And this institute really is all about bringing together the best scientists and the best clinicians so that we can work together and ultimately develop new diagnostics and new therapies. So I was recruited here last year as part of the phase 1 development of the institute, and we were officially opened in June of last year by His Royal Highness the Duke of York. And we had the opportunity to show him around our labs and our clinical space. And I told a few jokes. And we're now well into fundraising for phase 2 of the institute development, and this is going to allow us to build research capacity even further. So I think there are some really exciting developments going on that will ensure that cutting-edge research informs our medical decision-making here. So with that, I'd like to acknowledge everybody in my team for all of their hard work and dedication, and particularly to acknowledge the funding bodies without whom none of this would be possible. So I hold an MRC Senior Fellowship, and I've also received grant funding from Diabetes UK, JDRF, Type 1 Mission, BBSRC, and the Wellcome Trust. So thank you very much. I'd be happy to take questions.